Welcome. You are joining us for a deep dive that is going to take a fundamental piece of neuroscience and totally deconstruct it.
Right. Something you likely thought was completely settled science.
Exactly. I mean, if you follow neurobiology or even just the broader cultural conversation around mental health, you have definitely encountered the narrative around serotonin.
Oh yeah, the happy
A
chemical.
happy chemical, right? It gets branded as this molecule of good moods, the ultimate target of all these pharmacological interventions, and it's generally just treated as this unidirectional lever for well-being. But today, we are tearing that misconception down to the studs.
We really have to because that characterization is a profound systemic reductionism.
Because serotonin is not just some simple mood booster. It is actually the brain's ultimate structural architect, and it's highly sophisticated real-time manager for how you process, endure, and adapt to stress.
Yes, and when we actually interrogate the neurobiological literature, we find a system that acts as both a foundational structural engineer during embryonic development and a dynamic, high bandwidth network operator in the adult brain.
Which is wild to think about.
It is. We are going to examine the physical architecture and the real-time electrophysiological function of this system. We are specifically going to focus on how distinct receptor populations, namely the 5-HT1A and 5-HT2A receptors, literally dictate whether your brain defaults to patiently enduring a stressful environment.
Which we classify as passive coping.
Right, passive coping, or whether it radically destabilizes its own networks to adapt to new realities, which is active coping.
And the mission of this deep dive is to map that exact dichotomy for you. We're exploring the cutting-edge neuroscience of passive and active coping mediated by serotonin and its specific receptors.
And we are pulling from a massive stack of source material today.
A huge stack. We're going to trace the lineage of this research, starting with foundational developmental biology papers from 2003, moving through comprehensive anatomical reviews, and culminating in the absolute state-of-the-art. I'm talking brain-wide transcriptomic neuroimaging studies, including a landmark 2022 paper from Nature Neuroscience.
And those incredibly complex optogenetic imaging updates from 2024 using 11.7 Tesla MRIs on awake subjects.
Yes, we are going to break down the mechanics, the methodology, and the massive implications of these studies.
Because the literature we are covering today represents a true paradigm shift in systems neuroscience. We're moving past the era of viewing neurotransmitters as just localized signals.
Right.
We're stepping into the era of understanding them as global state modulators. By analyzing structural changes, network connectivity, and large-scale regional activation, you are going to gain a rigorous mechanistic understanding of the biological scaffolding that underpins your own behavioral resilience.
Okay, let's unpack this. I want to start with the sheer architectural scale of the serotonergic system, because the raw numbers present an immediate paradox.
The numbers are staggering when you really look at them.
They are. Consider the global human brain. We are talking about roughly 86 billion neurons in this densely packed, incredibly complex matrix. But the actual number of neurons that synthesize and release serotonin,
It's tiny.
it's microscopic in comparison. In the human brain, we are looking at roughly 350,000 cells. In a mouse model, it's about 26,000. That is an astonishingly tiny fraction of the total cellular population.
Mathematically, it's equivalent to having a few dozen engineers attempting to actively manage the routing protocols for the entire global internet in real time.
Exactly. Yet their influence is absolute.
And that paradox of scale is what makes the system so structurally fascinating. A 2010 paper by Yves Charnay and Lucien Leger, published in Dialogues in Clinical Neuroscience, it's titled Brain Serotonergic Circuitries. This paper provides the precise anatomical mapping that explains how this is physically possible.
Right, because these 350,000 cells aren't just scattered randomly.
No, not at all. They are heavily centralized, exclusively clustered deep in the brainstem within the rough nuclei, which anatomists divide into the B1 through B9 cell groups. Charnay and Leger detail how the rostral group, which is the B6 to B9 clusters comprising the dorsal and median raphe nuclei,
Was heading upward.
exactly. They project an impossibly dense, highly collateralized web of axons upward into the forebrain. Simultaneously, the caudal group, the B1 to B5 cells, projects downward to innervate the spinal cord and peripheral systems.
I want to pause on that term, highly collateralized, because I think it's easy to gloss over.
It's a crucial concept.
In standard neuroanatomy, we often visualize a neuron as having a single axon that reaches out and connects to a specific dendrite on another neuron, like a discrete point-to-point wire.
A simple circuit.
Right. But collateralization means these serotonin axons are branching off thousands of times. A single neuron sitting in the brainstem might be sending branches into the visual cortex, the prefrontal cortex, and the hippocampus simultaneously.
The metabolic burden on that single cell body to maintain miles of axonal wiring is immense.
Just to keep that one cell alive while it manages that much physical real estate.
The metabolic cost is staggering, which indicates how evolutionarily vital this architecture is. And what we must understand is that this massive, sweeping projection system doesn't just spontaneously organize in adulthood.
It's there from the beginning.
Very beginning. It is acting as a fundamental developmental signal almost immediately after conception. Klaus-Peter Lesch and Jonas Wader published a critical paper in 2012 in the journal Neuron titled Serotonin in the Modulation of Neuroplasticity and Networks.
A great paper.
They synthesize a wealth of data to show that before serotonin ever acts as a neuromodulator of mood or behavior, it acts as a morphogen. It physically moderates neurogenesis, controls the branching of other non-serotonergic axons, and dictates synaptogenesis across the developing brain.
I always think of it like the original construction foreman of the brain. The scaffolding has to be laid down before the building can function.
That's a perfect analogy.
According to a 2003 paper by Patricia Gaspar and colleagues in Nature and Neuroscience reviews, titled The Developmental Role of Serotonin, and this is corroborated by another 2003 paper by Vitalis titled Serotonin Homeostasis and Serotonin Receptors as Actors of Cortical Construction, these cells are generated remarkably early, very early. In the embryonic development of mice, serotonergic neurons are birthed between embryonic days 10 and 12. In humans, this corresponds to the very first month of gestation. These tiny clusters in the primitive brainstem start firing out pioneer axons before the cortex even really exists in its final laminated form.
And the physical mechanics of how those pioneer axons navigate the embryonic brain are deeply elegant. Gaspar and Vitalis detail a highly specific interaction in the marginal zone of the developing cortex. The serotonin axons physically interact with Cajal-Retzius cells.
Now, for those who might not know, Cajal-Retzius cells are transient developmental neurons. They don't stick around.
Right, they have one main job.
Their primary job is to secrete an extracellular matrix protein called reelin, which acts as a chemical gradient to guide the radial migration of newborn cortical neurons.
So the serotonin axons are effectively communicating with these Cajal-Retzius cells, modulating their activity to ensure that the 6 layers of the mammalian cortex are stacked in the precise, correct order.
So if that serotonin signaling is disrupted during that specific embryonic window, say due to genetic variations or maternal environmental factors, the implications aren't just a temporary chemical imbalance. You're talking about a physical alteration in the cortical columns.
Exactly. Gaspar's work implies that transient developmental disruptions in serotonin homeostasis lead to permanent architectural changes in how the brain is wired, which of course cascades into permanent alterations in adult behavior.
It's baked into the hardware.
That structural permanence is exactly the point. The physical routing of the network is determined by early serotonergic gradients. But even in the adult brain, the way these highly collateralized axons release their payload challenges are classical models of neurotransmission.
This introduces the concept of vesicular synergy, right?
Yes. Detailed deeply in a 2012 paper by Patricia Gaspar and colleagues titled Probing the Diversity of Serotonin Neurons, and further explored in the 2010 Charnay and Leisure Review.
Here's where it gets really interesting. In a classic, simplified model of the brain, neurons follow Dale's principle, or at least a strict interpretation of it. One neuron, one neurotransmitter. A dopamine neuron releases dopamine. A GABA neuron releases GABA. So naturally, a serotonin neuron releases serotonin.
That was the assumption for decades.
But the physical reality at the terminal ends of these massive cortical projections holds a massive functional plot twist.
The twist is that a significant subpopulation of these serotonergic neurons natively co-express a protein called VGLUT3. That's the vesicular glutamate transporter 3.
Which changes everything.
It does. To understand the gravity of this, we have to look at the biophysics of a presynaptic terminal. When a neuron prepares to fire, it packages its neurotransmitters into vesicles, these small Lod spheres, using specific transporter proteins. The discovery of VGLUT3 in serotonin terminals means that these neurons are actively pumping glutamate, the brain's primary fast-acting excitatory neurotransmitter, into the exact same vesicles that hold serotonin.
Let's contextualize why that is so radical. Glutamate is the gas pedal of the brain. It triggers rapid, highly localized ion channel openings that cause immediate depolarization. Serotonin is a slow-acting neuromodulator that initiates complex, cascading G-protein intracellular signaling. Packing them into the same lipid vesicle is essentially binding a high-explosive charge to a slow-burning thermite reaction.
That's a vivid way to put it.
When that vesicle merges with the membrane and dumps its contents into the extracellular space, you are getting an immediate excitatory spike followed by a prolonged state modulation.
And the thermodynamic implications of co-packaging are also vital here. The presence of glutamate inside the vesicle actually alters the electrochemical gradient across the vesicular membrane.
Because glutamate has a charge.
Yes. This shift in the gradient drastically increases the efficiency of VMAT2, the vesicular monoamine transporter 2, which is responsible for loading the serotonin. In simple terms, moving glutamate into the vesicle chemically forces more serotonin into that same vesicle.
It's supercharging the payload.
This synergy isn't just a quirk. It is the physical prerequisite for a phenomenon known as volume transmission.
We need to dissect volume transmission because this fundamentally breaks the standard model of how we imagine brain communication. If classical synaptic transmission is two people whispering across a tiny table in a crowded room, like a highly secure point-to-point transfer of information across a microscopic synaptic cleft, how does volume transmission alter that dynamic?
Volume transmission shifts the paradigm from a private whisper to a localized broadcast. In classical neurotransmission, the receptors are located directly across the synaptic cleft, mere nanometers away.
Right across the gap.
But because of vesicular synergy, these tonically active serotonin terminals can release massive, concentrated payloads of serotonin that simply diffuse through the extracellular fluid. The serotonin molecules drift away from the release site and bind to extrasynaptic receptors located on distant dendrites, cell bodies, and even passing glial cells over a wide physical radius.
So a single serotonin axon branching into the prefrontal cortex isn't just activating the 10 neurons it physically touches. It is essentially flooding a three-dimensional pocket of cortical tissue, altering the firing thresholds of potentially thousands of neurons simultaneously. It's shifting the ambient neurochemical weather of that specific brain region.
And that ambient weather shift is what sets the stage for our behavioral state. This diffuse volume transmission is how a tiny cluster of 350,000 cells in the brainstem can rapidly dictate whether a creature weighing 80 kilograms decides to fight, flee, freeze, or adapt.
Which transitions us directly into the functional core of this deep dive, the coping dichotomy.
Yes, the behavioral output of all this architecture.
To make this concrete, I want you to evaluate a scenario. Imagine a state of acute, inescapable adversity. It could be a chronic, high-stress professional environment where you have zero autonomy, or a sudden, catastrophic personal failure. When that ambient stress signal floods your system, your biology is forced to execute a strategy. Do you hunker down, moderate your internal anxiety, and simply tolerate the toxic environment, waiting for it to pass? Or do you undergo a radical behavioral shift, destabilizing your current life to aggressively seek a completely new environment?
That choice is governed by the bipartite model of serotonin function. It's a framework brilliantly synthesized by Robin Carhart-Harris in a 2017 paper published in the Journal of Psychopharmacology titled Serotonin and Brain Function, A Tale of Two Receptors.
A hugely influential paper.
Absolutely. Carhart-Harris aggregates decades of receptor pharmacology to propose that serotonin transmission enhances two distinct, structurally mediated adaptive responses to adversity. The routing of the volume transmission we just discussed hits two primary receptor subtypes, the 5-HT1A receptor and the 5-HT2A receptor.
The tale of two receptors. Let's start with the default state. Passive coping, governed heavily by the 5-HT1A receptor. We define passive coping as the biological capacity to tolerate a source of stress, to actively suppress the internal generation of anxiety, and to promote psychological patience.
Exactly.
When you are stuck in grinding traffic or dealing with an infuriating bureaucratic delay and you manage to not scream at the steering wheel, that is your 1A network doing the heavy lifting. But how does binding a molecule to a receptor actually generate the psychological experience of patience?
To answer that, we have to look at the electrophysiology. Lesh and Waiter's 2012 Neuron paper details the exact intracellular cascade. The 5-HT1A receptor is a G-protein coupled receptor that is profoundly inhibitory. When serotonin binds to the 1A receptor, it initiates a signaling cascade that physically opens a specific ion channel in the neuron's membrane called the GIRK2 channel, the G-protein-gated inward rectifying potassium channel.
Let's visualize the physics of that channel. A resting neuron is already negatively charged inside compared to the outside. Potassium ions, which are positively charged, are highly concentrated inside the cell. When the 1A receptor forces that GIRK2 channel open, the potassium ions rush out of the cell, following their concentration gradient.
And that efflux of positive charge drops the internal voltage of the neuron even lower. It hyperpolarizes the cell membrane, moving the neuron's electrical state further away from the threshold required to fire an action potential.
The neuron is effectively silenced.
Silenced. Now, the location of these receptors dictates the behavioral outcome. The 1A receptors exist in two strategic populations. First, they sit directly on the serotonin-producing cells in the rethe nuclei as somatodendritic autoreceptors.
Which acts as an incredibly elegant negative feedback loop. When the rephe nuclei start firing intensely and the local concentration of serotonin gets too high, the serotonin binds to its own 1A autoreceptors, opening the GIRK2 channels, hyperpolarizing the very cell that released it and forcing the system to cool down.
It prevents neurochemical runaway.
Precisely.
But the second population, the postsynaptic heteroreceptors, is where the behavioral experience of coping is generated. These 1A heteroreceptors are massively concentrated in the limbic system, particularly in the hippocampus and the amygdala, which are the primary structures for processing fear, threat detection, and emotional memory.
Okay, wait. If the 1A receptor's job is to hyperpolarize and silence neurons, and it is densely packed into the amygdala, isn't that just a biological mechanism for emotional blunting? If we are silencing the threat detection center, aren't we just ignoring the stressor?
If we connect this to the bigger picture of evolutionary survival, emotional blunting in the face of an inescapable stressor is highly adaptive. Active coping, fighting, or fleeing requires massive caloric expenditure and carries a high risk of injury or death.
True.
If a stressor is mild, or if the organism determines it lacks the power to change the environment, the most mathematically viable survival strategy is to minimize metabolic output and endure. By hyperpolarizing the overactive circuits in the amygdala during a stressful event, the 1A receptor network physically enforces a state of stress moderation.
It forces you to chill out so you don't exhaust yourself.
Exactly. It facilitates what behavioral neuroscientists call waiting impulsivity, the biological capacity to delay gratification or tolerate aversive stimuli without engaging in destructive, impulsive actions.
It's the neurobiological equivalent of weathering the storm in a reinforced bunker.
I like that.
But eventually, a bunker isn't enough. If the stressor escalates from a mild annoyance to an existential threat, say the environment becomes physically dangerous, or a psychological model of the world completely collapses, passive coping becomes a fatal strategy. You can't just hyperpolarize the amygdala and ignore a predator.
Oh, you definitely can't.
This is where the system flips, and we enter the domain of active coping, mediated by the 5-HT2A receptor.
Active coping requires the organism to completely change its relationship to the stressor. It demands heightened environmental sensitivity and a massive increase in cognitive plasticity to identify new solutions. If the 5-HT1A receptor is a heavy blanket dampening the electrical noise of the limbic system, the 5-HT2A receptor is a lightning rod placed directly over the executive centers of the brain.
To understand the 2A receptor, we just reverse the biophysics of the 1A. It is fundamentally excitatory.
Very excitatory.
Yeah. When serotonin binds to the 2A receptor, instead of opening potassium channels to silence the cell, it activates an entirely different G protein cascade, typically the GQ pathway, which ultimately leads to the closing of potassium channels and the release of intracellular calcium. This traps positive charge inside the cell, depolarizing it and moving it much closer to the threshold of firing.
A comprehensive 2015 review by Leiser and colleagues in Pharmacology and Therapeutics, titled Serotonergic Regulation of Prefrontal Cortical Circuitries, maps the density of these 2A receptors.
And they are in a completely different spot than the 1A.
Yes. Unlike the 1A receptors localized in the limbic threat centers, 5-HT2A receptors are intensely concentrated in the prefrontal cortex, the area responsible for complex planning, executive control, and cognitive flexibility. They are expressed on the dense apical dendrites of the main excitatory pyramidal neurons, as well as on local inhibitory interneurons.
So if the 1A is the shock absorber for a bumpy road, the 2A is the navigation system rerouting you entirely?
Exactly. Carhart-Harris proposes in his 2017 model that when an organism faces critical, unmanageable adversity, the dorsal rapher releases a massive surge of serotonin that overwhelms the 1A passive coping threshold and heavily engages the 2A network in the prefrontal cortex.
It essentially forces the brain out of the bunker.
This mass depolarization opens what he terms a window of plasticity. It physically unwires rigid pre-existing synaptic connections, the heavily reinforced behavioral priors that are no longer working, and allows the brain to rapidly encode new strategies.
Wow.
And what's fascinating here is the sheer structural complexity of how these 2A receptors operate. They don't just act as solitary switches. We have to return to the 2010 Charnay and Lear paper, which highlights a phenomenon that completely blurs the lines between discrete neurotransmitter systems. Receptors in the lipid membrane can physically migrate and bind to one another to form complexes.
This is one of those concepts that forces you to rethink cellular biology. We're talking about dimerization.
Yes, the 5-HT2A receptors frequently form homodimers, two 2A receptors structurally linking together to alter their binding affinity. But far more radically, they form heterodimers.
Meaning they bind with a completely different kind of receptor.
Yes. In the human cortex, the 5-HT2A receptor can physically bind to a mediatropic glutamate receptor, specifically mGLR2-2, to form a 5-HT2-mGluR2 complex.
Let's break down the mechanics of that heterodimer. You have a receptor designed to read the ambient serotonin volume, and it is physically welded to a receptor designed to read the ambient glutamate volume.
They undergo allosteric interaction.
Right. When serotonin binds to its side of the complex, it changes the physical shape of the glutamate side, altering how the neuron responds to excitatory signals. It is literally integrating two massive streams of chemical data to calculate the precise level of cognitive flexibility required for the environment.
This raises an important question. We have mapped the chemistry. We know that 1A hyperpolarizes the limbic system and 2A depolarizes the prefrontal cortex. But how does that ionic shift actually compute thought?
How does it scale up?
Right. How does a localized change in membrane voltage translate into a highly synchronized complex behavioral strategy across billions of neurons? To bridge that gap, we must translate this chemistry into electricity. We have to look at the genesis of brainwaves.
Exactly. Because the brain is a massive, oscillating electrical symphony, and the serotonin system is operating the mixing board. Two brilliant papers provide the structural blueprint for this. A 2013 paper by Puig and colleagues titled Serotonin Modulation of Cortical Neurons and Networks in Frontiers in Integrative Neuroscience, and a highly specific 2017 paper by Othlingham and colleagues in eLife titled Serotonin Enhances Excitability and Gamma Frequency Temporal Integration in Mouse Prefrontal Fast-Spiking Interneurons.
Othillingham's 2017 paper is crucial for understanding active coping. When you are engaged in intense cognitive processing, when you are solving a novel problem, adapting to a sudden threat, or actively recalculating your reality, your prefrontal cortex generates high-frequency electrical rhythms known as gamma oscillations.
Fast waves.
Yes, these waves cycle between 30 and 100 hertz. Othillingham's team demonstrated that these gamma rhythms are not generated by the primary pyramidal cells themselves, but are instead tightly orchestrated by a highly specialized subpopulation of cells called fast-spiking interneurons, or FSIs.
Fast-spiking interneurons are the metronomes of the cortex. They are typically parvalbumin-positive cells, and their architecture is unique. They don't project long distances. Instead, a single FSI wraps its localized axons around the cell bodies of hundreds of local excitatory pyramidal neurons.
They are heavily enmeshed in the local circuit.
Because they fire incredibly fast and release inhibitory GABA, they act as an electrical brake. They simultaneously silence hundreds of pyramidal cells for a fraction of a second, release the brake so all the pyramidal cells fire at the exact same millisecond, and then slam the brake back on. That synchronized rhythmic firing of excitatory cells is the gamma wave. It is the physical manifestation of focused attention.
And Puig's 2013 paper maps exactly how serotonin controls this metronome. Serotonin acts directly on these fast-spiking interneurons to modulate the temporal integration of the network. Puig's data shows a direct push-and-pull dynamic mediated by the 1A and 2A receptors.
And this is where the coping dichotomy hits the electrical layer.
Exactly. If researchers pharmacologically block the 5-HT1A receptors, the amplitude and power of the gamma oscillations actually increase. Conversely, if they block the 5-HT2A receptors, the gamma amplitude decreases.
Let's synthesize the gravity of that mechanism. The dorsal raphe nucleus dumps serotonin into the prefrontal cortex via volume transmission. If it binds primarily to the 1A receptors on these metronome cells, it dampens the gamma frequency. It reduces cognitive hyperfocus, promoting the passive coping strategy of enduring and waiting.
Right.
But if the stress is critical, and the ambient serotonin volume is high enough to massively engage the 2A receptors, it ramps up the FSI firing rate. It drives intense, high-amplitude gamma oscillations, physically forcing the prefrontal networks to synchronize and process data at maximum capacity to execute an active coping strategy.
And this structural rhythm management is not confined to high arousal states of wakefulness. Puig's 2013 paper explores the serotonergic modulation of the brain during sleep and deep anesthesia, states characterized by slow wave oscillations around 1 to 2 hertz.
So very different from gamma.
Extremely different. These slow waves represent a fundamental state of network rest, defined by massive cortex-wide alternations between UP states, where neurons are synchronously depolarized and active, in down states, where the network is deeply hyperpolarized and silent.
It's the brain breathing, essentially,
Yeah.
and electrical inhalation and exhalation.
The 2013 data demonstrates that physiological release of serotonin from the dorsal raphe physically shapes this breathing. Even during these low arousal states, serotonin rapidly promotes the initiation of UP states and significantly truncates the duration and amplitude of DAO-1 states. The serotonergic system is constantly structurally managing the electrical architecture of the brain across all states of consciousness, optimizing the network for whatever behavioral state is required.
So what does this all mean when we zoom out from the microscopic level? We've traced the system from embryonic pioneer axons navigating the marginal zone to vesicular synergy and volume transmission, to the competitive electrophysiology of the 1A and 2A receptors, to the real-time tuning of gamma oscillations. But how do we visualize this across the entire human brain simultaneously?
This brings us to the recent technological renaissance in neuroscience. The 2022 to 2024 literature shifts this from isolated cellular mechanisms to globally mapped networks.
We're entering the domain of macroscopic network validation.
Yes. I want to highlight a landmark 2022 paper published in Nature Neuroscience by Piergiorgio Salvan and his colleagues, titled Serotonin Regulation of Behavior via Large-Scale Neuromodulation of Serotonin Receptor Networks. This paper is a masterclass in methodological integration because it merges deep spatial transcriptomics with global functional imaging.
The methodology here is staggering, so let's carefully break down transcriptomic neuroimaging. Salvan's team didn't just look at fMRI blood flow. They utilized the Allen Brain Atlas, which contains microscopic voxel-by-voxel maps of gene expression derived from postmortem human brains.
Incredible resource.
They isolated the exact spatial gradients of mRNA that code for the construction of specific serotonin receptors. They effectively created a three-dimensional heat map of where every single 1A, 2A, 2C, and 3A receptor is genetically programmed to be built across the entire human cortex.
And then came the neuroimaging integration. They took those precise transcriptomic maps and overlaid them onto resting state functional MRI scans gathered from 812 living, healthy human subjects provided by the Human Connectome Project.
812 subjects. That's massive for an fMRI study.
It's huge power. They employed complex mathematical models to correlate the spatial density of the receptor RNA with the temporal fluctuations of the BOLD-D signal, the blood oxygenation level dependent signal, which is our proxy for neural activity.
By doing this, they proved something revolutionary. They demonstrated that the physical distribution of these receptors creates highly specific, coordinated, large-scale functional networks, which they termed SRNs, serotonin receptor networks. Serotonin isn't just washing over the brain and causing random activation. The structural architecture of where the receptors are physically located dictates a massive, synchronized global response.
To establish causality, to prove that serotonin release actually drives these specific networks, Salvan's team conducted parallel experiments using optogenetic fMRI in mice.
Tell us about the optogenetics.
Optogenetics involves introducing a viral vector into the mouse brain that inserts a light-sensitive ion channel, typically channel rhodopsin 2, specifically into the serotonergic neurons of the dorsal raphe. They then implanted a fiber optic cable directly into the brainstem. While the mouse was in the fMRI scanner, they pulsed blue light at 20 hertz, forcing the referrafin neurons to fire synchronously and release a massive wave of serotonin across the brain.
And the BOLD-D response they captured physically validated the receptor logic we've been discussing. The activation of the exact same serotonin source in the brainstem resulted in deeply heterogeneous network responses. The amplitude of the networks defined by the 5-HT1A and 5-HT2C receptors significantly increased. But crucially, the network defined by the 5-HT3A receptor actually decreased in amplitude.
And the divergence of the 3A network is a beautiful confirmation of the underlying biophysics.
Right.
Unlike the 1A, 2A, and 2C receptors, which are metabotropic G protein-coupled receptors that initiate slow, cascading intracellular changes, the 5-HT3A receptor is an ionotropic receptor.
Which means it works totally differently.
It is a pentameric ligand-gated ion channel. When serotonin binds to it, it acts immediately, like an excitatory glutamate receptor opening a pore for sodium and calcium. Salvin's optogenetic data proves that the slow, modulatory networks and the fast, ionotropic networks respond to the same ambient serotonin volume with completely distinct, spatially segregated Boldy dynamics.
But Sellin's scheme didn't stop at just mapping glowing networks in mice. They brought it back to the 812 human subjects to tie these serotonin receptor networks directly to observable human personality traits and behavioral models. And their findings perfectly validate Carhart-Harris's 2017 theories on coping mechanisms.
They absolutely do. They isolated the functional connectivity of the 5-HT1A networks, specifically analyzing how strongly the 1A expression mapped to activity in the amygdala.
And the psychometric data correlation was striking.
Very striking. They found that individual variations in the functional connectivity of the amygdalar 5-HT1A SRN directly predicted a behavioral metric called delayed reward discounting.
Delayed reward discounting is the clinical measure of waiting impulsivity. It quantifies an individual's capacity to forego a small, immediate reward in order to tolerate a delay and receive a much larger reward later.
The famous marshmallow test, essentially.
Yes, if your 1A network is structurally dense and functionally highly connected in your amygdala, you possess a massive biological capacity to hyperpolarize your threat centers, suppress the urge to react impulsively, and patiently endure a delay. Salvin functionally proved in hundreds of living humans that the 5-HT1A receptor is the physical substrate of passive coping.
Furthermore, they found that the connectivity of the 5-HT4 serotonin receptor network, particularly spanning the parietal cortex and the insula, directly dictated an individual's baseline reward responsiveness.
Yeah.
The structural layout of your neurochemistry is heavily dictating your psychological disposition.
And the methodological rigor of this field continues to accelerate. The 2024 literature refines these models even further by removing historical confounds.
I want to point to a brilliantly engineered 2024 paper by Hamada and colleagues titled Optogenetic Activation of Dorsal Raphe Serotonin Neurons Induces Brain-Wide Activation. The critical advancement in this paper lies in the physical hardware and the state of the subjects. They performed optogenetic mapping using an 11.7 Tesla MRI on awake mice.
We have to emphasize the physics of that imaging hardware. A standard clinical hospital MRI operates at 1.5 or 3 Tesla. The magnetic field strength dictates the signal-to-noise ratio and the spatial resolution. An 11.7 Tesla magnet is an absolute behemoth.
It's unbelievable.
It allows researchers to capture microscopic hemodynamic fluctuations with incredible temporal fidelity. But the real breakthrough is the awake state of the mice.
The awake state is the defining variable that recontextualizes decades of prior research. Historically, optogenetic fMRI studies, due to the physical constraints of the machinery, were performed on anesthetized mice.
Because you can't have them moving around in the scanner.
Right. In those anesthetized states, forcefully activating the dorsal raphe typically yielded a net suppression of widespread cortical Boldy activity. This led to models suggesting serotonin's primary global role was generalized dampening. However, Hamada's 2024 awake state data proved that anesthesia was a massive distorting confound.
When the mice are awake, processing real-time sensory data, and maintaining active cortical tone, stimulating the dorsal raphe doesn't suppress the brain. It does the exact opposite. Hamada captured massive, positive, brain-wide, bold responses across widespread cortical and subcortical areas. The ambient weather shift of serotonin in a waking brain is a driver of global network activation.
And Hamada's mathematical analysis of that awake activation perfectly mirrors Salvin's 2022 SRN mapping. The intensity and spatial distribution of the positive bold response was highly predictable, based on the structural density and the opposing expressions of the 5-HT1 family, which exerts an inhibitory weight on the network, and the 5-HT2 family, which exerts a positive excitatory weight.
It's the dichotomy written large.
Yes. The complex dynamics captured by the 11.7 T magnet were essentially the real-time, macro-scale visualization of the 1A versus 2A coping dichotomy fighting for dominance across the cortex.
To bring this entirely back to the human experience, we look to another 2024 paper, a clinical study by Caldwell and colleagues, titled Direct Serotonin Release in Humans Shapes Aversive Learning and Inhibition. Using advanced imaging and pharmacological challenges during behavioral tasks, Caldwell confirmed in living humans that the deep anatomical networks Hamada mapped in mice are actively shaping how we inhibit our physical behavior when faced with aversive, highly stressful stimuli.
Colwell's work is the ultimate translational validation. It demonstrates that the theories of passive coping, waiting impulsivity, and behavioral inhibition are not abstract psychological constructs. They are grounded in the real-time biophysical release of serotonin acting upon highly specific, structurally ingrained transcriptomic networks. Your ability to navigate a hostile environment is fundamentally a function of how your 1A and 2A receptors are spatially distributed and how effectively they modulate your gamma oscillations.
What an incredible intellectual journey this literature provides. We started by looking at a tiny cluster of 350,000 cells in the brainstem, watching their embryonic pioneer axons literally lay down the tracks of the developing cortex, guided by the chemical gradients of Cauchy-Retzius cells.
We unpacked the thermodynamic plot twist of vesicular synergy, where glutamate and serotonin are forced into the same lipid vesicle to enable massive extrasynaptic volume transmission.
We then explored the bipartite behavioral model. We mapped how the 5-HT1A receptor acts as a biological shock absorber, opening GIRK2 channels to hyperpolarize the amygdala and facilitate the passive endurance of stress.
And we contrasted that with the 5-HT2A receptor, the cognitive lightning rod that depolarizes the prefrontal cortex, heavily modulating fast-spiking interneurons to drive the high-frequency gamma oscillations required to radically rewrite your behavioral strategies.
And we culminated with the breathtaking macroscopic data. Salvin's 2022 integration of the Allen Brain Atlas with human fMRI to prove the existence of serotonin receptor networks, and Hamada's 2024 Awake 11.7 Tesla imaging, proving that the physical density of these opposing receptors dictates the global hemodynamic state of the brain. The knowledge contained in these papers is not academic trivia.
No, it is a profound mechanistic understanding of the physical scaffolding that literally builds our resilience, our patience, and our adaptability. It is the structural architecture of human endurance. This raises an important question, and perhaps it is the most vital question we can ask when we synthesize this much data regarding structural plasticity. We know the 1A network drives our capacity to patiently endure, and the 2A network explicitly unwires priors to allow for radical behavioral adaptation.
Here is the thought I want you to mull over as you step back into your day. If your inherent capacity to cope with life's most intense, inescapable adversity is mathematically dictated by the structural density and the functional connectivity of these serotonin receptor networks, and if we know for a fact that engaging the 5-HT2A receptor actively opens a physical window of plasticity that can alter structural synapses,
Right.
to what extent can we intentionally, consciously reshape our own receptor maps? Could deliberate, intense psychological interventions, focused cognitive exertion, or guided neurotherapies over time physically redraw the transcriptomic networks in our prefrontal cortex and amygdala? Could we, through sheer force of directed plasticity, give ourselves a permanent biological upgrade to our baseline coping mechanisms? The neuroscience suggests that the architecture for that change is built directly into the system, just waiting for the right signal. Thank you so much for joining us on this deep dive. Keep learning, keep questioning, and we will see you next time.